Many scientific studies, like these, which are attempting to discover the function of opiates and opiate receptors in normal brains, compare a normal control group to an experimental group which has undergone some sort of a treatment. In these cases, there are mutations in genes, and so one can observe what happens when a gene is absent and thus get a role of what it does in normal individuals. However, one must be careful to minimize the variables so that the only variable between two groups is this experimental variable, the absence of this mu opiate receptor in this case. Researchers, however, must make sure that the only difference between the groups that they are comparing is this experimental variable. So, for example, if one was testing the role of opioid receptors in repressing pain, say upon being exposed to heat, one must make sure that your experimental mice have the same ability to perceive heat as the normal mice. If there are two differences between the populations, one, the ability to detect opiates and thus suppress pain, but then two, the ability to detect heat. If these two variables differ, then the resultant difference in behavior could be attributed to either one. In the same way, if researchers were hoping to demonstrate that normal mice use their opiates and opiate receptors to suppress pain in a way that the mutant mice cannot with their non-functional receptors, one would potentially need to show that there is no difference in the ability to perceive whatever stimulus was causing the pain, such as a specific chemical. Only if both mice could perceive this chemical equally would then a difference be because of the opiate receptor not suppressing pain. The same concern could be potentially present if one were going to examine the brain differences in mutants which no longer expressed the mu opiate receptor. However, there are other opiate receptors, not just mu. One assumes that turning off one gene does not affect the expression of others, but that is an assumption. It is possible that, say, the kappa opiate receptor expression would change in the brain of a mouse that wasn't making the mu opiate receptor. One would need to control for that, because if not, then any difference seen between the control and experimental group, what would it be caused by the lack of the mu opioid receptor, or because of the change in the expression of the kappa opiate receptor? And so therefore, research papers very often do experiments to demonstrate that the control and experimental groups are comparable in all ways except for the experimental variable which is being tested.